This is a video to help you through the activity on structure-function relationships and protein denaturation. Let's take a step back and appreciate how the structure of a protein is suited for its individual function. We'll do this by comparing three different proteins that have very different functions. Notice how the shape and structure of the protein is suited for its function. The first example is the potassium ion channel. Potassium is an ion which is transported through the membrane either through facilitated diffusion or active transport, depending on the situation. Either way, it needs a protein to get through the phospholipid bilayer. The function of the potassium channel is to transport potassium through the cell membrane. And so it has an arrangement of amino acids so that nonpolar amino acids border the phospholipid fatty acid tails. The part of the channel that's facing the water solution on the outside and on the inside contain hydrophilic or polar side chains. In order for the ion to get through the channel, the center of this protein channel must also be polar. This protein forms a tunnel through the cell membrane where potassium can flow through. If you think of it as a donut, where the cell membrane is here and the donut is allowing a hole through the cell membrane, the outside of the donut has nonpolar residues and the center of the donut would have polar residues to allow the potassium ions through. If you look very carefully at this picture, there are three little green dots, and these represent potassium ions inside the potassium channel. Hemoglobin is another protein, but it has a very different function. It needs to be soluble inside the cytoplasm of cells, which means that it contains polar residues on the outside surface. Hemoglobin's job is to carry oxygen, and so it also contains oxygen-binding heme groups. The word hemoglobin comes from the fact that it contains hemes, which are represented here in the ball and stick models on each one of the subunits of hemoglobin. Notice that this is a very different type of protein than the potassium ion channel because they have very different functions. Another example is collagen. Collagen has a very different function from the other two proteins. The job of collagen is to be strong and flexible. Collagen is made out of three alpha helix proteins that are wrapped around each other. The three proteins here are shown in an aqua color, and then two of them are shown in like a pinky and a purple color. And in this structure, the ribbon diagram is also surrounded by a space-filling diagram. Collagen is the protein that makes up connective tissue, and so it has to be long, strong, and flexible. This is a fibrous protein. Potassium channels allow transport of ions through a cell membrane. Hemoglobin needs to be soluble and it transports oxygen through the blood. And collagen needs to be strong and flexible, and it's what makes up the connective tissues in our bodies. These three proteins have very different functions, and their structure suits their function. The structure of a protein is essential to the function of the protein. You can learn more about these proteins by clicking on the links below, and please do so. The function of these proteins is called the biological activity, and when the proteins are functioning correctly, they're said to be biologically active. If the protein isn't functioning correctly, it's not biologically active. There are two things I'd like to point out that can change the biological activity of a protein. One is a change in the amino acid sequence. Let's look at hemoglobin as an example. There are two main features of hemoglobin. One is the fact that it must be soluble, and the other is the fact that it has to have a heme group which binds to oxygen. Oxygen binding is an important function of hemoglobin. But the other important function of hemoglobin is to be soluble in water. A mutation in hemoglobin can cause sickle cell anemia. It's actually the mutation of only one amino acid. It's the sixth amino acid on the protein chain, and it's an amino acid that's on the surface of hemoglobin. When the glutamate in position 6 of hemoglobin is mutated to a valine, it changes the structure and the function of the hemoglobin. The hemoglobin can still carry oxygen, but it changes the way it behaves. It's no longer completely soluble. Here's a cartoon version of what happens when glutamate 6 is mutated to valine. There are 4 subunits in each hemoglobin, and each one of them would carry the mutation. The mutation here is highlighted in green. When a mutation of this position occurs from glutamate to valine, you change a polar amino acid to a nonpolar amino acid. The nonpolar valine does not like to interact with water, and so it would change the way that this behaves. It's like the valine tries to hide from water, and what happens is that the proteins aggregate in solution. Focus right here and notice that there's a highlighted mutation between this hemoglobin and this hemoglobin. The proteins will aggregate to hide the mutation between them instead of allowing it to be exposed to the water. When this happens, it causes an aggregation. The aggregation will push the cell into a sickle shape, and this is what causes sickle cell anemia. The second thing that will cause proteins to become dysfunctional or will disrupt its biological activity is protein denaturation. Protein denaturation usually means a loss of activity. You see protein denaturation when you cook eggs. Here's a picture of a raw egg, and the solution inside is clear and viscous. This solution contains dissolved proteins, and many of them are globular proteins. This is a cartoon showing a bunch of globular proteins that are folded. We heat and denature these proteins, or basically we unfold them. In the folded protein, the outside surface is hydrophilic or polar, and the inside is hydrophobic or nonpolar. When they're denatured, the hydrophobic or nonpolar parts are exposed, and they end up making a network of denatured proteins. At the same time, you remove water. This causes an egg to become cooked, and you can see that it looks very different. Heat is just one thing that will denature proteins and cause them to become unfolded and inactive. On your activity is a list of things that will denature proteins: heat, acids and bases or changes in pH, or ionic compounds, reducing agents, heavy metals, or mechanical agitation. And there's also a list of the forces that each one of the denaturing agents will disrupt. These are all attractive forces or side chain interactions. These are all things that cause secondary and tertiary structure, and depending on the denaturing agent, it will denature different levels of protein structure. I'd like to point out that denaturation is simply unfolding of a protein, so it disrupts attractive forces or disulfide bonds between side chains, but it does not affect the peptide bonds that hold primary structure together. If primary structure is affected, then that means a hydrolysis reaction has happened, and this is called digestion. So please take a moment to consider the difference between digestion, which is cutting of the peptide backbone, and denaturation, which is unfolding of the protein and disruption of secondary, tertiary, and/or quaternary structure. Thanks for watching this video about protein structure-function relationships and denaturation of proteins. If you have any questions, please post them in the comments, and if you like this video, please let me know by giving me a vote.